Good morning, everybody, and welcome to Unacademy. This is your educator Surbhi, and I welcome you all in today's class about microbial metabolism, part 2, where we are going to discuss about bacterial or microorganism metabolism. Let me first confirm whether I am audible and visible or not. So let's start with today's session. And let me first introduce myself. So this is Surbhi Singh, and I am PhD student at IIT Guwahati, and I have qualified CSIR NET examination in the year June 2019. Okay, so now coming to next thing, that is what you will get in this platform of Unacademy. तो ये Unacademy का platform आपको provide करवाता है daily live classes, and in these live classes you can have a one-to-one interaction with your educator. You can ask your doubt in the live classes. ठीक है, इसके साथ ही आप यहाँ पे टेस्ट सीरीज अटेंड कर सकते हो, क्विजेज अटेंड कर सकते हो, बहुत सारे वेल स्ट्रक्चर्ड कोर्सेज को आप बेनिफिट ले सकते हो. यहाँ पे जो कोर्सेज होती हैं, यहाँ पे आपके कंप्लीट सिलेबस को कवर किया जाता है. मतलब जो भी आपका चैप्टर है उसमें कॉन्सेप्ट ए से लेके कॉन्सेप्ट जेड तक सारे कॉन्सेप्ट्स और सारे पॉइंट्स को यहाँ पे कवर कराया जाता है. इसके साथ ही अनअकैडमी आपको अनलिमिटेड एक्सेस प्रोवाइड करवाता है टू ऑल द कंटेंट जो कि यहाँ पे अवेलेबल है. ठीक है, लाइव वीडियोस अगर आप नहीं देख पाते हो तो उन सब की रिकॉर्डेड वर्जन आपके लिए हमेशा अवेलेबल रहते हैं. So with these 4 benefits of daily live classes, live test series and quiz session, structured courses and unlimited access, you can get these benefits from Unacademy. Now coming to next thing, that is Unacademy platform also has free classes जिसको हम बोलते हैं special classes. ये जो special classes होती हैं ये सबके लिए free होती हैं. You do not need any kind of subscription in these classes. आप क्लासेस ये फ्री की क्लासेस अटेंड कर सकते हो. इसके लिए सिर्फ आपको अनअकैडमी लर्निंग ऐप डाउनलोड करनी है, और लर्निंग ऐप डाउनलोड करने के बाद आपको वहाँ पे अपनी प्रोफाइल क्रिएट करनी है, एंड यू हैव टू सेट योर गोल. So if you are preparing for CSIR NET examination, you can set your goal at CSIR-UGC NET, और उसके बाद आप स्पेशल क्लासेस के सेक्शन में डिफरेंट एजुकेटर्स का नाम सर्च कर सकते हो, या फिर उनके स्पेशल क्लासेस का शेड्यूल आप देख सकते हो. तो आप मेरी स्पेशल क्लासेस भी अनअकैडमी के ऊपर फॉलो कर सकते हो. एंड ऑन द अनअकैडमी लर्निंग ऐप में क्लासेस लेती हूँ फ्रॉम 8:30 PM. आज मेरी क्लास है फ्रॉम 8:30 PM, जहाँ पे हम गेट बायोटेक का पेपर डिस्कस करने वाले हैं. ठीक है, हम गेट बीटी का पेपर डिस्कस करने वाले हैं एट 8:30 PM. And if you are interested in Unacademy subscription, so you can use my referral code, that is Surbhi01, and ये कोड यूज़ करने से आपको मिलेगा 10% ऑफ. ये मेरा रेफरल कोड है SURBHI01. इसको यूज़ करने से आपको मिलेगा 10% ऑफ. और कोई भी प्लान, लेट से आप 12 मंथ का प्लान परचेज करना चाहते हो, तो ये प्लान आफ्टर हैविंग अ 10% ऑफ डिस्काउंट यू विल गेट दिस प्लान ऑन 10,395. सिमिलरली आप 12 मंथ्स का प्लान भी परचेज कर सकते हो अकॉर्डिंग टू योर शेड्यूल जब आपको एग्जाम देना है. सो दैट्स ऑल अबाउट द इंट्रोडक्शन पार्ट. नाउ कमिंग टू द टॉपिक. तो हम लोग आज बात करने वाले हैं माइक्रोबियल मेटाबॉलिज्म. सो एज वी हैव डिस्कस्ड ऑलरेडी इन द यस्टरडेज क्लास दैट what is metabolism. Metabolism is the sum of the chemical reaction in a organism. मतलब ओवरऑल एक ऑर्गेनिज्म जो भी केमिकल रिएक्शंस अपने अंदर परफॉर्म करता है टू ब्रेक डाउन द फूड, टू एक्सट्रैक्ट एनर्जी फ्रॉम दैट फूड, and to utilize these energy. So all these chemical reactions are collectively known as metabolism, and metabolism is further categorized into catabolism and anabolism. So anabolism and catabolism. Catabolism is a process of energy releasing. So catabolism में basically क्या होता है? Let's say when you talk about the catabolism of glucose, so glucose is broken down into simpler form, that is pyruvate. Now again this pyruvate is broken down into more simpler form of acetyl coenzyme A. Now this acetyl coenzyme A will enter into a TCA cycle and finally it will be converted into CO2. मतलब ग्लूकोज में जो C6 होता है वो रिलीज होता है in the form of CO2. So this is the process of catabolism, and during this process large number of ATP is generated. So it is a catabolism process. Now anabolism is the process where this ATP that is produced from the catabolism reaction, now it is used to synthesize different molecules like proteins, nucleic acids, cell organelle, okay, proteins, enzymes and all that thing, gene expression. So this entire process is catabolism, and the later part where ATP is consumed is the anabolism. So basically catabolism is the energy releasing process and anabolism is the energy using process. Okay, so in this diagram let's say यहाँ पे क्या हो रहा है, कैटाबॉलिज्म. ठीक है, यहाँ से केमिकल एंड लाइट सोर्स जो भी है वो एंटर करा एंड process of catabolism will start. Now during the catabolism you will have energy production and this energy can be used for biosynthesis. So this process is called as anabolism, जहाँ पे अमीनो एसिड्स, न्यूक्लिक एसिड्स, प्रोटीन्स के सिंथेसिस के लिए अगर एनर्जी यूज होगा तो इट इज एनाबॉलिज्म. Now some amount of energy will also be used for motility, for transport of nutrients, for cell maintenance. So energy that is being synthesized from the catabolism ये दो तरीके से यूज होगा. And during this catabolism a lot of waste products are generated. For example, during the catabolism of glucose, waste product is CO2. So waste products like acids, alcohol, CO2 are generated that can be eliminated out of the body. ठीक है, तो ये है हमारा पूरा का पूरा माइक्रोबियल मेटाबॉलिज्म एंड ओवरऑल प्रोसेसर. So for any metabolic reactions first the organism needs to catabolize, and catabolism के लिए उसको क्या चाहिए, nutrient चाहिए. So nutrient is the first requirement. Now this nutrient needs to be taken in the cell. So transport across the membrane, nutrient cell के अंदर आ गया. Now it is catabolized and catabolized into smaller and simpler products and finally it will result in the generation of ATP. So now this part is the catabolism part, and now after the ATP generation this ATP is used to synthesize several molecules. So first of all it will synthesize small molecules like amino acid, nucleotides. Now these amino acids and nucleotides will polymerize to form macromolecules like proteins, like DNA, like RNA. And finally these macromolecules assemble to form different organelles like ER, Golgi, mitochondria. So this is the overall energy flow that goes in a cell during its metabolism: nutrient uptake, metabolism, synthesis of small molecules and macromolecules, and then their association in an organelle. So now the next thing is the growth requirement. So microbial growth conditions. So for any microorganism to grow, it basically require macronutrients, micronutrients, growth factor and environmental factors. So environmental factors की अगर हम बात करते हैं, so they can be temperature, pH and oxygen. ठीक है, मतलब optimum temperature होनी चाहिए जहाँ पे microorganism grow कर सके, and that is around 30 to 37 degrees Celsius for mesophilic organism. Then pH should be optimum like 6 to 8 pH होना चाहिए. ऑक्सीजन लेवल, अगर ऑर्गेनिज्म ऑक्सीजनिक है या फिर एरोबिक है तो वहाँ पे ऑक्सीजन अवेलेबल होना चाहिए. अगर ऑर्गेनिज्म एनारोबिक है तो वहाँ पे ऑक्सीजन क्या होना चाहिए, अवेलेबल नहीं होना चाहिए. So environmental conditions or environmental factors play an important role in the microbial growth condition and microbial growth status. So microbial or microorganism require about 10 elements in large quantities because they are used to construct carbohydrate, lipid, proteins and nucleic acid. So basically 10 elements are required in large quantity and they are the macronutrients, and several other elements are needed in very small quantity because they are the part of certain enzymes, they are part of certain cofactors. So almost 10 elements are basically macronutrients, and rest can be micro, or there can be certain growth factor associated. Okay. So microbial nutrition. So nutrients are the substances in the environment that are used by the microorganism for catabolism and anabolism. So basically nutrients are the one that participate in the process of catabolism, and we have a macronutrient. That means it is required in large amount, including carbon, oxygen, hydrogen, nitrogen, sulfur and phosphorus. CHOPNS. ठीक है, कार्बन, हाइड्रोजन, ऑक्सीजन, फॉस्फोरस, नाइट्रोजन एंड सल्फर. सो दीज आर द वन दैट आर मैक्रोन्यूट्रिएंट, चॉपन्स. ठीक है, ये होते हैं मैक्रोन्यूट्रिएंट्स, एंड दे आर द कंपोनेंट ऑफ कार्बोहाइड्रेट, लिपिड, प्रोटीन एंड न्यूक्लिक एसिड. ठीक है, then some amount of potassium, calcium, magnesium and iron. These are also macronutrients. So 6 plus 4, there are total 10 macronutrients you have for the microbial, you require for the microbial growth. So it is carbon, hydrogen, oxygen, phosphorus, nitrogen, sulfur, potassium, calcium, magnesium and iron. ठीक है, तो ये हो गई फर्स्ट कैटेगरी मैक्रोन्यूट्रिएंट्स की, एंड रिक्वायरमेंट होती है टोटल 10 मैक्रोन्यूट्रिएंट्स की. नाउ माइक्रोन्यूट्रिएंट्स. तो माइक्रोन्यूट्रिएंट्स आर दोज व्हिच अ माइक्रोब रिक्वायर्ड इन वेरी स्मॉल अमाउंट. ठीक है, फॉर एग्जांपल इट कैन बी कॉपर, molybdenum, जिंक, एंड दे आर ऑफन रेफर्ड एज ट्रेस एलिमेंट. ठीक है, तो ट्रेस एलिमेंट्स कौन कौन से होते हैं हमारे पास? जिंक, कॉपर एंड मोलिब्डेनम. ठीक है, these are the trace elements and they are mostly required for the activity of enzymes, and they are commonly called as cofactors. ठीक है, so one is the macronutrient, next one is the micronutrient. Okay, next is growth factors. So growth factors can be amino acids, growth factors can be purine and pyrimidines, and they can be vitamins. So amino acids are needed for protein synthesis, fine. Purine and pyrimidines are needed for nucleic acid synthesis, fine. And vitamins are the small organic molecules that usually make up all or part of an enzyme cofactors, and they are usually required in very small amount. ठीक है, तो यहाँ से अमीनो एसिड आपको लार्ज अमाउंट में चाहिए, प्यूरिन पायरिमिडीन लार्ज में चाहिए, बट vitamins are the growth factor that are usually required in very small amount or trace amount. Okay, next topic is uptake of nutrients, where we are going to see how nutrient is being transferred from the extracellular space or from external environment inside a cell. The nutrient molecule frequently cannot cross the selectively permeable plasma membrane through passive diffusion and must be transported by one of the three major mechanisms involving the use of membrane carrier proteins. So usually जो nutrients होते हैं, nutrients are large macromolecules, so they cannot passively cross the plasma membrane, so they require specific transporters molecule. And specific mechanism की अगर हम बात करें, so in case of protozoans that are unicellular organism, the food uptake is basically being done by the process of phagocytosis, and in most of the microorganism, absorption occurs through the direct absorption from the membrane. Okay, and next is different types of transport. So let's have a quick review about different types of transports. So passive diffusion. So in case of passive diffusion, a few substances such as glycerol, basically substances that are lipophilic in nature, So a few substances like glycerol, water, oxygen can cross the plasma membrane by passive diffusion. Passive diffusion is the process in which molecules will move from a region of higher concentration to one of the lower concentration as result of random thermal agitation. ठीक है, तो due to the random movement of particles from an area of higher concentration to an area of lower concentration is known as passive diffusion. And basically it doesn't require any carrier protein, it doesn't require any energy consumption. So it is the simplest form of transport जो कि है passive transport. Okay, next is facilitated diffusion. So the facilitated diffusion, the rate of diffusion across the membrane is greatly increased by the use of carrier proteins. तो अगर आप नॉर्मल सिंपल डिफ्यूजन की बात कर रहे हो तो डिफ्यूजन इज बेसिकली अ स्लो प्रोसेस, बट व्हेन यू यूज अ स्पेसिफिक कैरियर प्रोटीन फॉर द ट्रांसपोर्ट ऑफ मॉलिक्यूल्स, so they are the facilitated diffusion method, and these carrier proteins are permeases which are embedded in the plasma membrane. And since the diffusion is aided by a carrier molecule, it is called as facilitated diffusion. So first one is the simple diffusion जहाँ पे there is no carrier protein involved, but in case of facilitated diffusion, carrier proteins are involved, and these carrier proteins will simply transport several molecules from their higher concentration to lower concentration without the use of any amount of ATP. So basically facilitated diffusion depend करेगा किस पे कि how many carrier proteins are there. Okay. So अगर हम बात करें सिंपल डिफ्यूजन की, तो सिंपल डिफ्यूजन प्रोसेस विल सिंपली डिपेंड ऑन द कंसंट्रेशन ग्रेडियंट, and the facilitated diffusion, it will depend on concentration gradient as well as your carrier protein concentration. So because here there is a dependency on the carrier protein, so यहाँ पे हमें जो ग्राफ मिल रहा है, it has a plateau phase, means at the highest transport rate, when all the carrier proteins are saturated, the rate will become constant. यहाँ पे आने के बाद जब सारे के सारे carrier proteins यूज हो गए होंगे या फिर सैचुरेट हो गए होंगे, उस पॉइंट पे क्या होगा, आपके सारे के सारे जो कैरियर प्रोटीन जब सैचुरेट हो गए होंगे, एट दिस पॉइंट द रेट ऑफ ट्रांसपोर्ट ये हो जाएगा कांस्टेंट. ठीक है, so the rate of facilitated diffusion increases with the concentration gradient much more rapidly, and at lower concentration gradient the amount of molecule that are diffusing is very low. So basically the diffusion occur from higher concentration to lower concentration, and it required several carrier proteins and permeases which are embedded in the membrane. So basically these transporters are embedded in membrane, and each protein carrier molecule is highly selective and will transport only closely related solutes. मतलब अगर एक कैरियर मॉलिक्यूल A है तो वो सिर्फ सिमिलर काइंड ऑफ मॉलिक्यूल्स को ही ट्रांसपोर्ट करेगा. नेक्स्ट इज अ मॉडल ऑफ फैसिलिटेटेड डिफ्यूजन. So basically this is the transporter molecule, and the membrane carrier can change conformation after binding an external molecule. So during this process, at the outer surface a molecule will bind, and the binding of the molecule to this transporter will induce certain conformational changes in this transporter, and this conformational change will allow the opening of this transporter in the cytoplasmic site, and now in the cytoplasmic site the molecule is delivered, and again it will undergo a conformational change, and again it will undergo the process of loading. So here there is loading, then there is a process of unloading, then again loading. So this loading unloading process is repeated until a concentration gradient exists. So the membrane carrier can change the conformation after binding to an external molecule, this process, and subsequently releases the molecule on the interior side. It is then returned to the outward orientation, and again loading will occur. Because there is no energy input molecule, transport will continue until the concentration gradient exists in this membrane transport molecule. Next thing is the active transport. An active transport is basically the transport of a solute or any molecule to a region from low concentration to high concentration or against the concentration gradient with the use of metabolic energy in the form of ATP. So basically active transport occurs from lower concentrated region to a higher concentrated region, and it is a energy dependent process. Here, okay. Next is the proton gradients. So proton gradients की अगर हम बात करें and how does transport occurs? So basically transport can be symport, that means linked transport of two substances in the same direction. Sim means same direction. So symport where two molecules are transported in the same direction. अगर दो molecule inward आ रहे हैं तो दोनों inward आएंगे, अगर दो molecule outward जा रहे हैं तो दोनों outward जाएंगे. So it is a symport transport. Next is antipode transport, and here the transport involves linking the transport of two substances in opposite direction. Means if this is the plasma membrane, this is the transporter, so one molecule will be transported out and another molecule will be bring in inside the cytoplasm. Unipode, that involves transport of only one molecule, either inward or outward. So sympode, antipode and unipode, these are the three type of transport mechanism that usually occurs across a membrane. So let's have a review. So first we have studied what? Simple diffusion process. Then after diffusion we have studied facilitated diffusion. Okay? Then facilitated diffusion is followed by the third category, that is active transport. And active transport is then followed by the fourth one, that is group translocation. So group translocation में क्या होता है? A process in which a molecule is transported into the cell while being chemically altered. मतलब अगर एक molecule A है, इसको अगर membrane के across transfer होना है, तो during the transport this molecule A should be chemically altered. The best known example of group translocation system is the phenol phosphoenol pyruvate sugar phosphotransferase system, PTS system. ठीक है? P for phosphoenol pyruvate, then phosphotransferase system. During this PTS transfer system, this system transports a variety of sugar into prokaryotic cell while simultaneously phosphorylating them using PEP as phosphate donor. So in this case, different sugars are being transported across the membrane by the process of phosphorylation. So it basically uses phosphoenol pyruvate as a phosphate donor, and PEP will donate its phosphate to the sugar molecule. Now sugar plus phosphate will be transported across the membrane. So during this process, PEP plus sugar in the extracellular phase will combine. Now this PEP will donate its phosphate to the sugar and PEP get converted into pyruvate, and now this sugar phosphate will be transported inside. So basically this PTS system is based on the phosphorylation of sugar molecule for the transport mechanism, and the component is divided into different parts, like it involves a PEP as a donor,
Hello, good morning all.
okay, sorry for the inconvenience. So we are talking about the group translocation, and during the translocation this phosphoenol pyruvate system So this system basically uses the phosphoenol pyruvate as phosphate donor, and it uses a specific set of enzymes, and these enzymes are, first is the E1 enzyme, that is enzyme 1, then a protein folding chaperone, that is a heat stable protein, and the E1 is a cytoplasmic component. Then it uses other components like E2, and this E2, so E2 can be of different subtypes. So E2 can be like you see, E2 can be a E2A, that is a cytoplasmic and soluble complex. E2A, that is a cytoplasmic and soluble complex. Then E2B, and E2B is a hydrophilic, but it is attached to E2C usually. Then E2C, and this E2C is a hydrophobic part that is embedded in the membrane. So when you see this figure, so this is the enzyme E1, that is a free enzyme, cytoplasmic enzyme, and it basically takes up the phosphate from PEP and convert PEP into pyruvate. Now this phosphate will be transferred from enzyme 1 to enzyme 2A, 2B, and finally to the enzyme 2C. Okay, and now this enzyme 2C will incorporate this phosphate to glucose molecule, and now glucose will be transported in the form of glucose 6 phosphate. So this is for the case of group translocation. Is it clear? Group translocation, this is important with respect to your syllabus. Let me first again tell you. So group translocation में क्या होता है, एक मॉलिक्यूल होता है जो कि ट्रांसपोर्ट होता है across the membrane, and during the transport of this molecule, this will undergo different chemical changes, and these chemical changes can be phosphorylation, it can be nitrosylation, it can be carboxylation. Okay, let's see how does this process happen. Okay, so during this process of transport, first of all, PEP will donate its phosphate to a molecule of enzyme 1. Now this phosphate will be donated to a sugar molecule, and now this sugar in the form of sugar phosphate will be transported across the membrane. So this phosphoenol pyruvate sugar phosphotransferase system is a very important class of enzyme that basically participates in the group translocation of several sugar molecules like glucose, like lactose, and different kind of molecules. And next thing in the transport system is the iron uptake. And iron uptake is basically associated with iron uptake. So iron uptake is basically a process that requires siderophores. So siderophores are Fe chelating compounds that are secreted by different microorganisms, and these siderophores, they are basically released by the microorganism in their vicinity or in the extracellular spaces. And in the extracellular spaces, these siderophores will complex with Fe+3 to form a soluble Fe+3 complexes, and these soluble Fe+3 complexes were then taken up by the use of active transport mechanism. So the first process is siderophores. They will form a complex with the Fe molecule in the Fe+3 configuration, and now this soluble Fe+3 siderophore complex will be transported with the help of active transport mechanism. So FeCD4 complex will first of all go and bind to siderophores receptors present on the outer membrane. Okay? They will first go and bind on the surface these Fe siderophores that are soluble complex, they will go and bind on the surface of siderophore receptors that are present on the outer membrane. Now, upon binding to the outer membrane, they will be transported with the help of a ATP binding cascade transporter, जहाँ पे एक ATP molecule use होगा और जो Fe+3 है वो transport हो जाएगा inside the membrane. So Fe plus enter into the pediplasm. Now they will be transported with the help of ABC transporters inside the membrane, that is in the cytoplasm. And now inside the cytoplasm, Fe+3 is reduced to Fe+2 for the process of utilization and uptake. So basically iron uptake is facilitated by siderophores. Siderophores will chelate the iron in Fe+3 configuration, and now siderophore plus Fe+3 become soluble and they will be transported with the help of active transport mechanism inside the cell. So this figure explains this. So S is for siderophore. Siderophore will make a complex with Fe, and now this Fe siderophore will bind to transporters molecules. So these are the transporters or receptors, and now they will be internalized by the ATP binding cascade transporter, that is ABC transporters. So that's all about today's lecture, and this is a review for like the conclusion from today's slide. So first there is 4 division we have discussed in today's class, that is passive diffusion, facilitated diffusion, active transport and group translocation. So in the basis of different categories हम इसको differentiate करेंगे. So carrier proteins की अगर हम बात करें तो passive diffusion, it does not require any carrier proteins, so none. In case of facilitated, yes. In case of active, yes. In case of group translocation, yes. ठीक है, इन 3 cases में there is requirement of carrier proteins, but passive diffusion does not need any kind of carrier proteins. Next is the transport speed. So here, because no carrier protein is involved, so the process will be slow, and these 3 are rapid. Against gradient, no. Passive diffusion is across the gradient, so no. This is also none, and these 2 are yes. ठीक है, active transport and group translocation, these 2 processes occur against the concentration gradient. And based upon the specificity, so passive diffusion doesn't have any specificity, but those transport mechanisms that require carrier proteins are specific. So facilitated diffusion, active and group translocation, they are specific in their action. Next is energy requirement. So passive diffusion will not require any energy. Facilitated diffusion, it will also, this is wrong, here it will be no. So facilitated diffusion also do not require any energy, but active and group translocation will require energy for their completion. So that's all about today's class, and thank you so much. If you have any doubt related to today's class, you can comment it in the comment box, and if you want to join our telegram channel, तो आप लेट्स क्रैक CSIR-UGC NET का टेलीग्राम चैनल ज्वाइन कर सकते हैं. उसका लिंक आपको डिस्क्रिप्शन बॉक्स में मिल जाएगा. एंड दोज हु हैव नॉट येट सब्सक्राइब द चैनल, आप सबसे पहले चैनल को सब्सक्राइब कर लो टू गेट एनी अपडेट. कोई भी अगर आपको अपडेट चाहिए तो चैनल को सब्सक्राइब करके आप हमारे क्लासेस की जो भी अपडेट्स हैं वो ले सकते हैं. एंड इफ़ यू आर नॉट येट यूजिंग अनअकैडमी, तो आप अनअकैडमी जल्दी से ज्वाइन कर लो. अनअकैडमी की फ्री क्लासेस आप ज्वाइन कर लो. आपको कोई सब्सक्रिप्शन लेने की ज़रूरत नहीं है. उसके लिए आप मुझे अनअकैडमी स्पेशल क्लासेस में भी फॉलो कर सकते हो. उसके लिए आपको बस अनअकैडमी लर्निंग ऐप डाउनलोड करनी है, वहाँ पे अपनी प्रोफाइल बनानी है, एंड यू हैव टू सेट योर गोल एट CSIR-UGC NET. और एक बार आप अपना गोल सेलेक्ट कर लोगे, देन यू कैन फाइंड माई प्रोफाइल देयर. ठीक है, स्पेशल क्लासेस में आप मेरी प्रोफाइल सर्च करना सुरभि कुमारी, आपको मेरी क्लासेस वहाँ मिल जाएंगी. आप मुझे एंड about my special class schedule. So today I will be taking a special class at 8:30 p.m. on GATE biotechnology previous year questions. So I hope everybody will join me at 8:30 p.m. on the Unacademy special classes platform. So that's all about today's class. Thank you so much and have a nice day.